Hello, everyone. My name is Cora Vacher. I'm Associate Director in the Strategic Marketing Team for Human Genetic at Oxford Nanopore Technology. I'm going to give you a quick introduction about how Oxford Nanopore Technology is working with a number of initiatives across the world and developing solutions to really enable the generation of global genetic data for much more equitable precision medicine in the future. So we all know understanding the genetic architecture of complex diseases has always been a numbers game, and it's now possible to get much richer insight on large group of participants with Oxford Nanopore sequencing. For example, long-read sequencing typically identify at least twice as many structural variants in germline if you compare to short-read sequencing. And remember, by sequencing the DNA natively, so direct sequencing, you do get simultaneously methylation data with your genomic information. So we're working with a number of projects across the world. We're actually finishing the work, the long read sequencing work with PRECISE. PRECISE is a project in Singapore that aims to sequence, using long read sequencing, 10,000 genome representative of the Singaporean population, so Malay, Indian, and Chinese. That group also is sequencing about 100 sample telomere to telomere to really improve the pangenome globally. The second project I wanted to showcase is the UK NHL bioresource. So that the group there, I put together 22,000 samples, and they've got sub-project within it. Some are rare disease, some have cancer, but a very large part is common complex disease, in particular working towards food disorder. And the input material kind of varies. So there's some blood, there's some saliva, etc. So it's not a completely homogeneous cohort. And on the opposite side, also in the UK, we have a very homogeneous cohort, the UK Biobank. And you may know that at the end of last year, we've announced that we were having a partnership with the UK Biobank to sequence, using long-read sequencing, 50,000 samples. When the data is available, that will make it the largest single cohort catalog for structural variation and methylation information. And by methylation, I mean 5-MC, but also 5-HMC. And the last project I wanted to showcase is the enrich card. And card stands for the Center for Alzheimer and Related Dementia. So as you can see, it's not the largest project. It's 4,000 samples. But the reason I think it's a very special project is because out of those 4,000 samples, the team has brain tissue for all of them and blood tissue. And it's quite rare to have so many brain tissue. And that team really is looking into the methylation pattern for Alzheimer and related dementia in those brain regions. So the sample in large cohorts and biobank are very varied. By that, I mean the input material. And even if you think just human cohorts, the most common are likely to be blood or dry blood, saliva, and buccal swab. But as I mentioned, we are seeing a fairly large cohort of tissue outside of cancer and cells that are really analyzed at scales. And of course, there is a really growing interest of application using cfDNA. And we do have supported solution and experience with many of those. So our P24, Promethean 24, and the 8-hour series, really the workhorse of large cohort sequencing. And it's really ideal for production sequencing because it can generate about 2,500 genome at 30x per year on a single device. And again, it does include methylation. I can't say that enough. You get your 5-MC and 5-HMC information at the same time that you get your genomic information. When it comes to data analysis, the Epitomy desktop application offers a very comprehensive analysis of the human genome on a single workflow. This really simplifies the investigation of human genomic variation, obviously including SNVs and structural variant, but also copy number and repeat expansion. And Epitomy really provides also secondary analysis, including annotation file and report for all the mentioned variants and SNV phasing block. And if you do want to do your visualization of the map read, obviously, you can, and the tracks, you can either do it within Epitomy or you can open your output file within your preferred genome browser. So I really hope I've shown you with about 5 minutes that the work that we are doing with those large cohorts is really enabling people to generate more insight, more multi-omic data, really much at scale. I wanted to finish with the disclosure slide. So Oxford Nanopore technology are research use only. The product label branded as Oxford Nanopore Diagnostics may be used for research use only or may be regulated in some jurisdiction, and please do check the individual product for labeling. Oxford Nanopore technology makes no endorsement of the material shown in the statement made. Thank you very much for your attention.
Thank you for inviting me to speak about the critical role of studying human genetic diversity in populations to achieve equitable precision medicine. So over the years, I often ask myself, how have human history and migration shaped our genetic diversity? As humans migrate out of Africa, they experience population bottlenecks, which alter genetic variation. These migrations also expose individuals to new environments where certain genetics variants confer adaptive advantages. For example, in the Himalayas, variants that enhance oxygen transport were positively selected. Another key question is, how does genetic variation influence disease and treatments? Some variants, once advantageous in Paleolithic diets, might now predispose us to metabolic diseases in modern society diets. And finally, how genetic diversity can help us to understand disease biology, identify new therapeutic targets, and validate biomarkers. I will argue that studying human genetic diversity isn't just an academic exercise. It's a powerful tool to deliver equitable precision medicine. Back in 2011, together with my colleagues Carlos Bustamante and Esteban Gonzalez Burchard, we published a commentary in Nature emphasizing the urgent need to increase diversity in genomics research. At the time, only 4% of individuals in the NHGRI GWAS catalog were from non-European ancestry, a glaring imbalance that risks perpetuating health disparities. We hope our call to action will catalyze change. This underrepresentation stems from both logistical and methodological challenges. It's easier to recruit participants in Europe and in the US, and it's easier to analyze relatively homogeneous populations. But such convenience comes at a scientific and ethical cost. While progress has been made, more recent data shows that only 9% of individuals in the GWAS catalog are from non-European origins. So it's clear that efforts to address this imbalance have fallen short. So what is at stake? Among many reasons to study genetic diversity, one stands out for precision medicine, drug discovery. Genetic diversity enables the identification of novel therapeutic targets, the validation of existing targets, and a better understanding of tolerability of drugs and biomarker performance. By including more diverse individuals, we gain access to a broader range of alleles shaped by natural selection in different environments, many of which are rare or population-specific. For instance, studying diverse cohorts increase our chances of discovering natural knockouts, individuals with loss-of-function variants that can inform drug development. A prime example is the gene PCSK9, a gene that has been implicated in cholesterol metabolism. This gene was first discovered in a participant in the Dallas Heart Study who had extremely low LDL cholesterol due to a natural knockout in PCSK9. Later, individuals with missense variants were also identified, and those had very high LDL cholesterol. So these insights led us to understanding the role of this gene in cholesterol metabolism, and then development of very powerful cholesterol-lowering drugs. This illustrates why genetic diversity is essential, not only for understanding disease biology, but also to enabling the development of new therapies. Let me now turn to one of the earliest applications of diverse population genetics, admixture mapping. In 2004, in collaboration with Steve O'Brien and David Reich, we developed an admixture map of microsatellites, markers, to discover disease genes in admixed populations. If the disease differs in prevalence in either of the source population of an admixed population, we can use this marker set to identify genomic loci associated with the disease. For example, that it tracks with African or European ancestry in a population of African American patients. A prototypical example is the Duffy locus, nearly fixed in African population due to its protective effect against malaria. And in the figure on the right, in the left, we show how this admixture map successfully highlights the locus, validating the approach. And this approach then later was used to identify other genes. As genotyping technology advanced, we began exploring the patterns of linkage disequilibrium across the genome. In 2005, we published the LD maps for chromosomes 6, 21, and 22, revealing recombination hot and cold spots, or haplotype bugs, as they will become more widely known, patterns that differ in intensity by ancestry. African populations show shorter haplotype blocks and lower LD, reflecting deeper population history and higher genetic diversity. In the figure on the right, we can see these maps for the different populations of the hap map, and we can see spikes on or steps on the map where recombination is higher, and plateaus when there's recombination cold spots. The green dots are the classical linkage disequilibrium map derived from crossovers in families. So we can see the pattern is very similar, but the map is longer in African Americans. This information became crucial for designing and interpreting genome-wide association studies later on. Later, as next-generation sequencing emerged, the 1000 Genome Project was launched to build a more complete catalog of human genetic variation. The initial panel of 1,000 individuals included the same samples from the HapMap project, Africans from Yoruba, collected in Nigeria, Han Chinese from Beijing, Europeans from the SEP collection collected in Utah, and Japanese collected in Tokyo. But my colleague Carlos Bustamante and I asked, is this truly representative of global human diversity? Can we learn important biology by including more diverse and, in fact, admixed populations? To exemplify this, in 2009, we sequenced the first Mexican and African American genomes and demonstrated that admixed populations offer unique insights. Our 2012 paper ultimately included 12 admixed individuals. And in this principal component analysis plot on the left, we can see the clusters of the subjects from the HapMap projects, and the distance here is the similarity, genetic similarity in these individuals. The ones with the black outline are the ones that we selected. So it's difficult to see, but we selected typically individuals in the middle of the admixture. In this study, we also introduced new tools to infer local ancestry across the genome. In other words, we can track haplotype segments derived from the two or more ancestries along the chromosomes. And in this figure on the right, we can see an example in chromosome 7 for the individuals on the top for Puerto Ricans individuals in our sample, Mexican individuals, and African Americans from the Southwest. We can see, for example, that the Mexican individuals have significant Native American ancestry in blue, that the African American individuals have significant African ancestry in the yellow, and in the top we can see that the Puerto Rican individuals, at least the two that we included in the study, have a mixture of European and African and a little bit of Native American. This data allows to reconstruct demographic histories. By analyzing track-length distributions, which reflect the impact of recombination over generations, we infer the timing and the extent of admixture events. The left panel shows the distribution of track lengths for African American individuals in our cohort, while the bottom graphic represents a demographic model illustrating the timing and source of gene flow, the pie charts indicating ancestry contributions, and the arrows at what number of generations in the past. This is a model that better fit the data and highlights major pools of African ancestry occurring about 7 to 8 generations ago. The right panel presents analogous information for the Mexican individuals in our cohort, indicating a significant pulse of Native American ancestry roughly 15 generations ago, which will end up in being about 50-50 European, Native American, and a small proportion of African ancestry, which we know this matches our historical records and what we know about these populations. Further, using methods like the pairwise sequential Markovian coalescence, PSMC, that was developed by Hen Lee and Richard Durbin, allows to infer population size in the past from just single individual if we want it. In these plots, we show the average for individuals in the different populations listed there, African American, Mexican, Puerto Ricans, Africans, and so on. And we can see that in the highlighted area, there is a population bottleneck with, in the past between 15,000 and 70,000 years ago, which is much more severe for individuals coming out of Africa. We were also able to apply this new method in our data, in this case on the segments that we derived from the admixed individuals. So we separated these segments in African ancestry, Native American ancestry, and European ancestry, and we can recapitulate what we knew about African ancestry and European ancestry, but for the first time we can see that Native American ancestry, which at the time has been fully unexplored, also suffered a very severe population bottleneck in the near past. So the 1,000 Genome Projects became the 2,500 Genomes Project and became an invaluable open access resource used widely for benchmarking technologies, ancestry inference, and a framework for disease studies and clinical genome interpretation. In that project, we also report the true extent of genetic diversity across different groups in the project. On the left-hand side, we can see this figure show the variant size per genome across different populations, showing that the African populations have the highest diversity, which they are on the right, and also South Asian populations are included there. And on the left-hand side, we can see that the populations from Northern Europe have the lowest level of diversity. And on the right-hand side, we can see, again, another PSMC analysis showing that all human populations suffer a very severe bottleneck 150 to 200,000 years ago. Now, this context laid out the ground for many studies, including our study, which is a recent work done on familial hypercholesterolemia, or FH. FH is a common but underdiagnosed disease, particularly in non-European populations. Its prevalence is about 1 in 200, 300, and typically only 1 in 200 individuals have been molecularly diagnosed. Nevertheless, the disease can be diagnosed clinically by the presentation of at least 3 of these physical findings, the more important which is the elevated levels of LDL-C early in life, for example, in young adults or even in children, and other findings such as tendinopathies, early onset cardiovascular disease, and family history of the disease. By now, we have good understanding of the role of various genes involved in FH and in the cholesterol metabolism, such as the LDL receptor, where 90% of the pathogenic mutations in FH cases are found, ApoB, and the PSK9 gene that I mentioned earlier. Molecular testing can confirm this clinical FH and can inform patient management and improve the therapy of these patients. So that's why it's important, the molecular diagnosis. For our study, we partnered with the Instituto Nacional de Ciencias Médicas y de la Nutrición of Mexico to study a cohort of 300 FH patients coming from 169 families. In other words, there were multiple individuals, sometimes from the same family. And we asked, what is the proportion of FH due to non-monogenic mutations versus the polygenic influence? Could structural variations play a significant role? And importantly, are there new genes to be discovered? Is there any other PCSK9 that could be gleaned from this unexplored population? This slide shows you the admixture that we found in this cohort. Each vertical line is one individual. The colors represent the ancestral genetic population on the genome. So we can see the proportions of the genome of the different ancestries. And what we see in pale blue is the ancestry from the Americas, from the Native American ancestry, and in dark blue from Europe, the European ancestry. So we can see that we have individuals in this cohort that range from mostly Native American ancestry to mostly European ancestry, which obviously makes things more complicated, but at the same time more interesting. And so what we first did was to identify pathogenic or likely pathogenic variants in this population, a fact that was complicated by the lack of information of some of these variants in this population. Nevertheless, we were able to identify significant amount of pathogenic variants in the LDLR receptor, ApoB, PSK9, LDL RAP1. Additionally, we also find mutations in other non-classical FH genes related to other dyslipidemias, such as ApoE, CREB2L3, and PLIN1. So these genes are assigned to different types of dyslipidemias, but clearly they are contribute to the burden of disease in this cohort. Interestingly, we identified two carriers of a duplication in a gene called PDZK1, a gene that encodes a scaffolding protein in the membrane that interacts with the HDL scavenger receptor, which caught our attention. We also identified, we look in the UK Biobank, and we identified 3 carriers of a duplication in this gene, also exhibiting high levels of LDL-C. So we're pursuing the hypothesis that this gene could contribute to the burden of FH. It's a story that we're following. In terms of molecular diagnostic, we achieved very high molecular diagnostic, 68% in the case of individuals from families, and 34% in single cases, still very high to the typical rates. So this population really represented an enrichment of cases, and tells about the advantage of dealing with registries or cohorts that are very well curated. 5% of the diagnostic were also copy number variants, a deletion in ApoB, LDLR, and a duplication of PSK9. Through the use of long-read nanopore technology, we were able to confirm some of these mutations. Here I'm showing at 14 kb heterozygous deletion of exons 13 and 14 of LDLR, fully phased. So we can see how we can easily resolve the breakpoints with this technology, which in some cases is difficult if there are repeats around the area. Short read technology is going to have problems there. Now, we aim to examine also what's the contribution, the polygenic contribution into this disease. In particular, there are individuals in the cohort that we could not diagnose with a pathogenic variant in the classical gene. So what's happening in these individuals? So to do that, we calculated polygenic risk scores of LDL cholesterol. The problem with polygenic risk scores, applying polygenic risk scores in these populations, is that it's known that these are mostly derived from GWAS studies, carried out in European populations, and they often poorly port to other ancestries. Alicia Martin in Carlos Bustamante lab time ago demonstrated that, by applying European-derived polygenic scores in African populations, there is either an under or over representation possible of the risk of the individual. So our solution was to derive methods that can adjust for these distortions. So we developed an ancestry-adjusted polygenic risk score that, after calculating the raw PRS, we recalibrate this score in the background of 1,000 individuals closer to the individual that we're analyzing in terms of genetic ancestry. We draw these from the Galatea Global Biobank, which includes all of the public data plus additional data that we've been collecting in diverse cohorts. And using that, we can recalibrate and obtain polygenic risk scores that are more reflective of the risk of the individuals. And to showcase these, here in the left-hand side, I show a plot where we calculated polygenic risk score for 4,000 models that we obtained in the PRS catalog. And the vertical lines represent the correlation between one model and European ancestry. So you can see, in general, there could be very high correlation between the score and European ancestry, which obviously is not a good property you want to apply into other populations. But after our procedure, the blue lines, most of this correlation has been eliminated. And on the right-hand side, if we see the area under the curve, which is a way to evaluate a model for its predictive ability, we can see that after the adjustment, the unadjusted scores is the orange plot and the adjusted is the blue plot, we improve in the predictability of these scores. Clearly, for a study, we selected a score with high predictability for LDLC, which is what I will show you later. So in this slide, I can show you what is the distribution of the scores. In particular, what's the proportion of individuals in the left-hand side above the 95th percentile for cases that are carriers of monogenic variants and cases that are non-carriers. And as compared to controls in the bottom, those controls are also from a Mexican population, but a different disease. So they are matched in terms of admixture. But we can see that even in the cases with monogenic variants, there is a higher proportion of individuals in the high end of the score, but clear that non-carriers have almost double. So polygenic variants are definitely contributing to the burden of disease in this population. And the right plot just shows how this distribution shift. You can see the entire distribution, how it's shifting in the cases and the cases non-carriers. Now, I'm going to turn into our latest study, expanding structural variation catalogs using non-read sequencing technology in populations that are underrepresented. In 2010, a large-scale project was launched by enrich LVI to investigate genetic factors in population of African descent. And this is an effort to try to improve the catalog of variation in African ancestry populations. So we decided to sequence 400 individuals from this collection that come from 18 populations of the African diaspora, including the Caribbean and South America, using Oxford Nanopore technology. And our goal was to expand the catalog of structural variations, which we know long-read will be more powerful for. Discover new structural variations that could be affecting pharmacogenes or disease-associated genes in these populations, and also leverage the long-read phasing to infer local ancestry segments, like I was mentioning some slides ago, to better reconstruct the demographic history of these populations. So this is a principal component plot where in colors we can see the population from the 1000 Genome Project. We can see Europe in one cluster, Africa another cluster, East Asia another cluster, the population from the Americas, in this case, in the 1,000 genome was Mexicans from LA. And in black are individuals of the populations that we're sequencing. So we can see they are closer to the African cluster, but they also represent other areas of genetic diversity that has been unexplored. You can see the populations names on the right-hand side. Some come from Barbados, Colombia, and so on. And we also include a few African populations that we have access to. With this technology, we were able to discover 31,000 structural variants per sample, which is double the number that you typically detect by short-read technology. On the right-hand side, we can see the top two plots show the deletions and insertions in the range of 50 to 1,000 base pairs, and in the bottom from 1,000 to 10,000 base pairs. And we can see the patterns that we expected. In other words, bigger events are less frequent because there's natural selection operating on them, but we also see the spikes of the ALU repeats and the line elements, giving us confidence this data is of high quality. We also perform benchmarking with reference samples to ensure this is high quality. As we compare this data to the recently started 1,000 Genome Project long-read sequencing with Oxford Nanopore, we compare this with the May 24 release. 60% of the structural variations that we found are novel, which clearly seems that we are contributing a good part of novel structural variation to the community. Over 200 of these structural variants impact 160 homing disease genes, which suggests that they may have, in some instances, clinical relevance or inform disease biology. This is an example. A deletion of 972 base pairs in exon 3 of the filaggrin gene. This is a gene that is involved in the integrity of the skin barrier, and exon 3 is where most of the non-pathogenic variants reside, but it's very highly repetitive in nature. You can see it has many units of this filaggrin repeat, which is the structural component that goes into the skin. And it's known that pathogenic variants, loss of function variants in this exon lead to increased risk of allergy, immune diseases such as atopic dermatitis, asthma, and others. The problem is that with this repetitive nature, this exon has been basically unexplored and is not typically tested because it's difficult to do, but with long-read technologies, we can easily see the deletion. There's still some other complexities here that we're exploring. We're going to be doing some assembly in some of these genomes to better clarify that. And what we're finding is that this is just one variant, but we find more frequent variation in this exon in terms of structural variants, sometimes deleting 1 or 2 units of this gene that is not understood what's the implication in terms of disease, but this kind of resource could be used for later on following in other studies. Now, in terms of pharmacogenetics, is another area where long-read shine. We can see that for some of these important genes like CYP2D6 that have a paralogous pseudogene, it's very difficult to sequence with short reads, but now with the long reads, we can see how we can immediately distinguish a deletion in one haplotype of the gene CYP2D6, which would be equivalent to the STAR5 pharmacogenetic allele as the nomenclature used in pharmacogenetics. And we have the other haplotype, includes a number of single nucleotide variants that are characteristic of the STAR29 allele. So allows to very clearly distinguish these alleles, but we also find novel alleles that are difficult to interpret because have been understudied. With the help of the Chinook algorithm developed by Oxford Nanopore, we were able to identify all these alleles in this gene, and using PharmCat, an algorithm developed by the PharmacoJKV group in Stanford, we were able to derive the metabolizers phenotypes. Here you can see the 2 arrows. We split our population in groups of high Native American ancestry or high African ancestry to compare with reference population from PharmaJKV. Interestingly, in the high Native American ancestry, we were unable to identify any subjects with ultra-rapid metabolizer, which suggests that maybe it's going to become harder to interpret genomes of other populations because there are novel alleles that have not been reported and cannot be characterized, which is the gray area where we cannot really understand those alleles. So this is an area of expanded study and long-read technology can be very helpful. In summary, for the CAAPA study, we found significant structural variation with long reads that is typically not described easily with short reads. In these underrepresented populations, this structural variation is novel, can impact genes. And in pharmacogenetics, using the bespoke method, we were able to call star alleles for CYP2D6, and we are also doing this for other pharmacogenes, and allows to study novel potential pharmacogenetic alleles in these populations. To close, studying human genetic diversity offers multiple benefits. It enhances our understanding of population history, improves variant interpretation in clinical settings, like in genetic testing, and unlocks new opportunities for drug discovery. So diverse genomes are not only essential for equity, they are powerful tools for scientific advancement. And with that, I want to thank my collaborators at Galatea Bio for the FH project, for the CAAPA project, including our Mexican collaborators of the Institute of Nutrition in Mexico that developed the FH registry, and the 1000 Genome Project for the background information that we all use. Thank you very much.